Hello everyone, next I will share the regulation of cardiovascular activity to you. The regulation of cardiovascular activities, including nerve regulation, body fluid regulation, and self-regulation, can not only maintain the relative stability of cardiovascular function activities such as normal heart rate, cardiac output, arterial blood pressure, and blood flow of various tissues and organs, but also in the internal and external environment of the body make corresponding adjustment when changing so that cardiovascular activities can adapt to the needs of changes in metabolic activities. Let's look at the neuromodulation first. Cardiovascular activity is regulated by the autonomic nervous system. The parasympathetic nervous system mainly regulates the heart activities, while the sympathetic nervous systems play an important role in regulation and activity of the heart and blood vessels. The heart is dual innervated by the cardiosympathetic nerve and the cardiovagus nerve. Excitation of cardiosympathetic nerve enhances the activity of the heart, while the excitability of the cardiovagus nerve inhibits the activity of the heart. Let's first discuss the cardiosympathetic nerve. In the middle lateral column of the thoracic spine cord, the ACH released from the axon and can activate the N1 cholinergic. The postganglionic neuron cell body of the cardiac sympathetic nerve is located in the stellate ganglion and cervical sympathetic ganglion. Its axon forms the postganglionic fibers innervating various parts of the heart. The postganglionic fibers of the cardiac sympathetic ganglion release norepinephrine and act on the myocardial cell membrane. The beta-1 adrenergic receptors on the upper part of the body cause increased myocardial contractility, increased heart rate, and increased conductor velocity. These effects are called positive inotropic effect, positive chronotropic effect, and positive allotropic conduction respectively. The ganglionic fibers of the parasympathetic nerve that innervate the heart wall in the vagus nervous trunk, and the cell bodies of ganglionic neurons are located in the dorsal nucleus and the suspicious nucleus of the medulla oblongata. The terminal of preganglionic neurons release ACH, which acts on a receptor in the body membrane of postganglionic neurons in the cardiac ganglion. The postganglionic nervous fibers of the vagus nerves mainly innervated the sinus node. Atrial muscles, the atriovascular junction, the atrioventricular bundle and its branches. While the innervation of the ventricular muscle is very small, the fiber ending of the cardiac vulgar ganglion also released ACH, which acts on the M-type cholinergic receptors of the myocardial cell membrane, causing weakened atrial muscle contractility, slower heart rate, and slower atrioventricular conduction velocity. Sexual degeneration, negative degeneration, and conduction of negative degenerations. In addition to the sympathetic and vagal nerve innervation, the heart has the following three kinds of nerves: peptidergic nerve fibers innervating the heart, efferent nerve fibers of the heart. Both the sympathetic and vagus nerves contain a large number of efferent nerve fibers. Its nerve ending mainly fuel chemical stimulation and mechanical strength stimulation from the heart. Tonus means that the nerves of muscle and other tissues maintain a certain degree of continuous activity. Cardiac sympathetic nerve and cardiovagal nerve are usually tonic, and both originate from the medulla oblongata cardiovascular center. The two function antagonize each other and regulate heart activity together. Sympathetic tension and vagus tension also change with the respiratory cycle. Vagus tension is lower during inhalation and sympathetic tension is higher. The nerves that innervate vascular smooth muscle are divided into two categories: vasoconstrictor nerve and vasodilator nerves. Except for capillaries, the blood vessel wall have smooth muscle distribution. Most of the vascular smooth muscles are only innervated by sympathetic vasoconstrictor nerve fibers, and only part of the blood vessels, except for sympathetic, in addition to the innervation of vasoconstrictor nerve fibers. It also receives the innervation of certain vasodilator nerve fibers. Vasoconstrictor nerve fibers are sympathetic nerve fibers, so they are called sympathetic vasoconstrictor nerve. And the transmitter released by the fiber end of the ganglion is norepinephrine. Vascular smooth muscle cells have two types of adrenergic receptors, alpha and beta 2. Norepinephrine and alpha receptors can cause vascular smooth muscle contraction, and beta 2 receptors can cause vascular smooth muscle relaxation. Norepinephrine has a strong ability to bind to alpha receptors, but a weak ability to bind to beta 2 receptors. Therefore, the main effect of sympathetic vasoconstriction nerve fibers is vasoconstriction. The distribution density of sympathetic vasoconstrictor nerve fibers in different tissues and organs is different. The innervation density in various blood vessels of the same organ is also different. Skeletal muscle vessels are not only innervated by sympathetic vasodilator nerve fibers, but also by sympathetic vasodilator nerve fibers. The postganglionic fiber and the released ACH, which acts on the M receptor in the vascular small muscle membrane, which can cause skeletal muscle vasodilator and increase skeletal muscle blood flow to adapt to the increased blood flow demand of skeletal muscle during exercise. Its effect can be blocked by the M receptor antagonist atropine. Sympathetic vasodilator nerve fibers do not have tonic activity at ordinary times and emit impulses when they are emotionally excited and have defensive reactions. The vascular smooth muscles of a few organs such as meninges, salivary gland, etc., not only receive the innervation of sympathetic vasoconstrictor nerve fibers, but also receive the innervation of parasympathetic vasodilator nerve fibers. Receptor binding can cause vasodilation and increase in local blood flow, but has little effect on the total peripheral resistance of the circumambulatory system. This type of vasodilator nerves usually has no tonic activity. In addition, there are two kinds of vasodilator fibers. One is the vasodilator fiber in the posterior root of the spinal cord. The other, peptide vasodilation nerve fiber. In the central nervous system, the concentration of the neurons related to the control of cardiovascular activity is called the cardiovascular center. The neurons that control cardiovascular activity are widely distributed at all levels, from the spinal cord to the cerebral cortex, and the medulla oblongata is the regulation heart. The hypothalamus, the most important central part of the cardiovascular system for vascular activity, also plays an important role in the regulation of the cardiovascular activity. There are close fiber connections and interactions between the cardiovascular centers at all levels, not only receiving various incoming information from the body and internal organs, but also receiving regulatory information from other parts of the center, and regulating cardiovascular activities through complex integration. Make cardiovascular activities adapt to the changes of interval and extraval environment and other function activities of the body. The medulla oblongata is the most basic center for regulating cardiovascular activities, as shown in the figure. It includes neurons in multiple locations with different positions and different functions. That's all for this lesson. Thank you for your attention. See you later.